Well, good afternoon. Thank you for the honor of being here. Thank you, the organizers, for allowing me to tell you something about what ideas we have in these topics and themes. I actually don't have a video to tell you something and for you to understand the Valle University. Valle University is the third biggest public university in Colombia. It has more than 30,000 students. 4,500 belong to the postgraduate programs, 252 academic programs, 160 are undergraduate, 450 groups of research, and more than 1,000 professors. Out of those 400 groups of research, I'm going to tell you a little bit about what we do in the research group that I lead and coordinate. This research group is a big group. It is formed by 3 labs. What I'm going to tell you about is what we do in the laboratory of microbiology, and probably in projects I'm going to show you in the end. I'm going to show you what my colleagues do in other laboratories because we have an internal transversal cooperation. Well, when I made this slide, I thought it was basic and probably it wouldn't be necessary to show, but after seeing the rest of the presentations here in this venue, I think it is important to tell you a little bit. It is about bioremediation. When we describe bioremediation, we know we have two concepts of bioremediation. The term bioremediation is born in this context, which is pollution of environment and everything related. Nevertheless, another context of bioremediation that does not appear associated to the term bioremediation and today is considered part of the study of bioremediation is the production of pollutants that we call in the end of a tube. We highlight characteristics of these two types of pollution. In one, we have diffuse contamination. They are problems with difficult control. Once the problem is onset, it is difficult to reverse it, as you saw in the morning talkings until the former presentation before me. The important thing we do is diagnose the problem, see the dimensions of the problem, and in the moment of control it is so costly that in most of the cases they leave it as such. So this is the worst case scenario. As you can see, much of this pollution comes also from all of these industrial and domestic processes of waste generation that afterwards reach there and leaches. A great part of agriculture occupies a great space of organic loads that are generated and go to the environment. This is an additional landscape. I'd like to tell you something about the story, the types of remediation, because they have developed individually. Fortunately, they go together, these two areas of work, and they have been covered and studied by specific groups of professionals. In one case, there's emphasis in microbiology and biology, and the other case, they are emphasized in engineering. These two engineering systems were worked as black boxes, and here we made more work and we supported more the development of microbiology and molecular biology. I'd like especially highlight this event here in the part of engineering for Colombia, and it is having a new standard in Colombia in the case of emissions of liquid waste. Standards until December last year covered 80% of removal. When covering 80% of removal many companies complied with standard, leaving a great amount of organic loads into the rivers and natural systems. This has changed. It means that it is likely, when we apply the new standards, that finally all of the groups that have been working via remediation and control of pollutants, probably they're going to find a better space of response and financing for projects, because all of the parts of pollution control is the black sheep. Nobody wants to invest. Having a treatment system for many companies is a problem, and investing in innovation for it is much more difficult. That's why, by following this context, here in this part, what I'd like to show is the difference of focus of research when we do a consultation on what they do in the rest of the world and what we do in LATAM. And much of what we do in LATAM, well, the consequences is that we are working very little in aspects related to molecular analysis, biosensors, and we are devoted to classic problems of pollution, which doesn't happen in the rest of the world, where there's better development in working with biosensors and molecular applications, tending to molecular solutions and nanotechnology, which is the reason for this event. One of the elements is the type of regulations and standards which make having our environments in extreme pollution. Another context that I'd like to give you into account is that waste, when we speak about waste, we speak about agricultural, industrial, municipal, and other industry waste related to different to agricultural industries that in general terms could be issued solid, liquid, and gas waste. And commonly, not always, actually there are cases where these are mixes of many pollutants, many types of organic and inorganic pollutants. This is a very complex reality. I've been working with my research group in pollution 20 years ago in several systems of treatment, solid, liquid, and gas waste, to give attention to the needs of the companies. But the concern is that with these new standards and the levels of pollution, we are finishing environment, and what can we do to convince entrepreneurs that they have to invest and do something different in favor of environment? The only solution is tell entrepreneurs that you can make money by giving treatment and caring for environment. This is a complex problem. So the proposal of solution is a proposal, a complex solution also. This is the model that we work, all of the research groups in my university. It is a complex model, and the interesting thing of this model, probably you know what we refer to a biorefinery, and the idea is that complement all of the biological processes that you could have in a biorefinery, not only for solid waste, but also liquid and gas waste, complemented with processes of advanced oxidation, adding advantages of advanced oxidation, which are clean processes or less polluting processes together with biological processes. Treatment methodologies that we use. Probably many of you working in bioremediation and in laboratories of microbiology, you acknowledge very well these methods. It is where you make a culture with a pollutant, a specific pollutant, and where you want to assess, and you choose the diversity. You choose some strains that could be promising, and then after you work with these strains, if they are not possible to grow, or microorganisms are not possible to grow, you can make clones and genetic modifications in order to study the possibilities of solution. But as you saw before, the problem is really complex when we have many pollutants, and when we're working in reactors, we find interesting situations, and it is that the proportion of microorganisms that we can observe directly could be lower, and the number of microorganisms doing a good job with so many pollutants necessarily is not a pure culture. That's why here, with the help, because we're in the School of Engineering, this is a very important aspect we have in our university, and one of our laboratories, the collaboration, give collaboration of engineering, and we have an easy access to work with reactors. And obviously, we have devoted our work and efforts into developing these methods of culture in reactors. This is a photo of microorganisms, animal microorganisms. They are non-cultivated, but cultivated in a strategy of a reactor. What is the difference of Petri or a reactor? Well, you have to model in the culture, in the continuous culture, you have to model each of the conditions that you need by the microorganism to grow in the way that you want it massively growing. This mechanism has never been used in a Petri or in a lab tube, but here we have reached 97% of purification of a culture based on this learning that we made in company of Mr. Mayet, Dr. Mayet, who's the pioneer in this type of method. We have developed what we have done so far. This means that the microorganisms are never cultured or isolated that way. Processes of selection are done directly in the reactor. And the idea is that we don't make transgenesis with this type of cultures, but we do culture engineering. What does it mean? We make a specific selection of microorganisms inside the reactor within the culture. So here we have some examples of what we have obtained. These are nitrogen-fixing bacteria, phenoculus, are obtained from wastewater from domestic use. These nitrogen fixers are highly resistant to ammonia concentrations, high ammonia concentrations. Additionally, we're not thinking only in the application of biomass on soils, but also apply in effluents, which could be rich in ammonia, and combine them with other type of biotechnology, such as in the case of algae, production of algae, which one of the limitations to implement in great scale is nutrients required and cost implied. But as you can see, these are the patterns of diversity that we obtain in several moments of the reactor, and some of the images of microorganisms. So far, we are just starting. There's a long way to go, and potentially there could be pathogens here, and one of the challenges is how to take out those non-convenient microorganisms in the culture without taking the others, knowing that many of those are not possible to culture. I leave you the inquiry if you can tell me something about it. This is the example for the obtaining biopolymers. I know this could be very daring, that any organic waste could be transformed in biopolymers, but I'm daring, and I think potentially doing the right oxidizing processes combined with anaerobic and aerobic processes in a biofactory, we could obtain all of the waste containing organic matters. We can turn them into biopolymers. This is an approach, economic approach. It doesn't have the whole detail, as mentioned before, but it is what we could recover economically. This chart is very good because this is the way as we talk to entrepreneurs and we try to convince them, to tell them this could make sense. What is ahead in the future? As I told you, we have to select only some microorganisms. We need to take out the pathogens without using any culture mechanism. We are starting using cytometry of flow in order to sort out those microorganisms and associate them as the way we need them in order to solve the problems And in other different projects, we are trying to develop materials with other colleagues of a group, and we expect to use several of our reactors, especially those nitrogen fixers, in order to obtain power, energy. If we can close the cycle by obtaining bioenergy through the metabolism of bacteria, those nitrogen fixers that generate a lot of electrons, we could have sustainability of a system like this. This is the great challenge: study the metabolites of the system, ecology of the system based on the genomics of these systems. And obviously, we're going to need much more information from several types of studies. Rapidly, they are telling me my time is over. Are some of the projects. This is the classic biorefinery project, but the idea is to have much more projects with much more elements in this type of biorefinery for solid liquid waste in the same process. This is a very important project for this type of model and system that we proposed, reactor development. This is the bottleneck we have of many laboratories, because one of these reactors is really expensive, and this is done in association with the Federal Polytechnic School of Switzerland, and there's much information online. We want to do it with open source, because in LATAM countries or anywhere they can reply the model, cover the model, and have versatile, cheap reactors in their laboratories without making big investments. This is another project where we are developing a biosensor to evaluate disinfection with a colleague in the United States. And this is a project that we're going to start with my colleague Howard Honka to use metagenomics for community analysis in reactors. These are other disinfection projects that we are carrying out with an Argentinian colleague, assessing disinfection and microorganism adaptation, comparison of patterns of antibiotic resistance compared to resistance to disinfection. Finally, my advertising. I'd like to greet my dear colleagues, Pablo, here present, Howard. They've been part of this initiative, which is the Latin American Association of Microbiology and Biotechnology, environmentally speaking. In this moment, we have it very forward. We have coordinators in Mexico, Argentina, Brazil, Uruguay, Peru, and we're looking for coordinators for Ecuador and other countries in LATAM. And we kindly invite you. In September, we're going to have the LATAM Microbiology meeting. We're going to have our first official meeting of the LATAM Association of Microbiology. We kindly invite you to participate in this venue. And those who do the job, to say, I do everything, well, they are the real workers. And this is an interdisciplinary work of microbiologists, chemical engineers, environmental engineers, biologists, biotechnologists, not only from Valle University, but also different universities from several countries. Thank you.